All right, welcome back. So in the second part of this lecture, we'll be looking at how these lipids are digested and absorbed and then delivered to the rest of the body. So hopefully most of this digestion is review, and we'll be going into a lot more detail about the actual absorption and delivery of lipids in this chapter. So of course, lipids are insoluble in water. They do not dissolve in water, right? Those tails repel water, in fact. And so we produce lots of different structures, or a few different molecules at least, that aid in the dissolving of these lipids or breaking apart of these lipids in order for us to digest them. And so throughout the digestive tract, anywhere where there is water, there's going to be very little digestion occurring of lipids. So I mentioned it before that we do have a tiny amount of salivary lipase, a small amount of lipase in our saliva. And so there is some chemical digestion of lipids happening in our mouth. And then some of that can continue in the stomach, but there is very little overall happening in these areas. So most of the digestion isn't going to happen until the small intestine. The main thing that is happening from chewing and the mixing of from our stomach into the gastric fluid is going to be mechanical digestion. But even that is just a small portion of the overall mechanical digestion that is going to happen to lipids. Most of both the mechanical and chemical digestion is not going to occur until the small intestine. And so this has an impact on the satiety value of fats too, right? Fats make us feel more full than the other macronutrients because they take the longest to digest, so they're taking up space, and they're the most energy dense. Right? So again, a tiny amount of lipase within our saliva, so there is some chemical digestion happening in the mouth. That lipase can continue in the stomach, but again, just a very small amount. Really, the main digestion of lipids is happening once they reach the small intestine. So in the small intestine, the gallbladder will release bile, which was produced by the liver, stored in the gallbladder, and then the gallbladder secretes that bile into the small intestine, and that bile will help with a majority of the mechanical digestion of our fats. And then our pancreas, which creates all of our different enzymes, the pancreas will secrete lipase into the small intestine, which will do the chemical digestion of our fats. So bile is essentially a salt. It is a small amino acid or a mineral attached to a cholesterol molecule, essentially a short fat. So as fat enters the small intestine, this molecule bile will be released from the gallbladder into the small intestine. This bile will then be able to interact with fat because it has a cholesterol portion and interact with water. And so this will help pull small pieces of that fat off. Right? So a few triglycerides at a time or many triglycerides at a time, but breaking down this large clump of fat into a bunch of smaller tiny balls. And that will allow for the enzyme lipase to then come in and chemically break apart those triglycerides. So bile disperses fat, mechanical digestion, right? It essentially is infiltrating this larger fat, like clump of fat. We'll start to break off some triglycerides into these smaller micelles, right? These small groups of fat. And this will allow for the enzyme lipase to bond with them and start chemically digesting those triglycerides. And we are digesting these triglycerides by separating two of the fatty acid tails from that glycerol head. So lipase will break these bonds. So thus two of the fatty acid tails are breaking off, and that leaves behind one fatty acid attached to the glycerol head, which is a monoglyceride. Again, mono meaning one, glyceride referring to the glycerol head. So lipase breaks off two fatty acids, leaving behind one monoglyceride. And so we then absorb fatty acids by themselves and monoglycerides. And so all of our long-chain fatty acids are going to be arranged as a molecule called a lipoprotein. Lipo referring to lipids, and protein referring to the proteins that are going to be surrounding the outside of it. And this will allow for those fats to then cross the membranes into our small intestinal cells, and then into our lymphatic system, and eventually into our blood, into our capillaries. So the largest and least dense version of these lipoproteins, we're going to be talking about multiple right? We've already referred to HDLs and LDLs, high density lipoproteins, low density lipoproteins. We'll have 4 different categories we talk about. And so we have a couple slides here talking about chylomicrons, which are the largest version of this lipoprotein and the least dense. So chylomicrons are a lipoprotein produced by the cells lining our small intestine. And so these are groups of chemically digested fatty acids, right, and monoglycerides surrounded by phospholipids, so essentially creating a membrane around them, and proteins. Those proteins are what are going to help these fats get to their target location and transport around our body. So as I said, this allows for the transport of lipids across our cellular membranes and keeps them together within our aqueous environments. Okay. So all of our fat is going to travel through the lymphatic system after it leaves our small intestinal cells. And so this will skip the liver and then transfer them directly into the bloodstream near the heart. So our carbohydrates and proteins, when they leave the small intestine, go straight to the liver, and the liver decides what happens to those things. Are they going to be stored? Are they going to be sent out into the circulatory system? Right? But lipids skip that. Lipids skip the liver on their first time traveling around the body, and they will be deposited near the heart. Right? Our short and medium chain fatty acids, however, so like I said, the long chain fatty acids are what are being arranged as lipoproteins. Most of our short and medium chain fatty acids can just be absorbed by themselves and absorbed much more quickly since they aren't being arranged into these lipoprotein structures. All right, so here's a look at what a lipoprotein looks like. So we have our triglycerides, or our fatty acids and monoglycerides and cholesterols, right? All of our different versions of lipids contained within this essentially a small cell or a vesicle. Right? So we have phospholipids surrounding the entire outer portion with membrane or proteins embedded into this membrane. So a lipoprotein has lipids contained within a phospholipid layer with proteins sticking out of it. And so it is the size and density of these lipoproteins that is going to determine how our body can use them or the health effects that they have. So here are the different versions of those lipoproteins that I was referring to. So we have chylomicrons, which are the largest and least dense. So these are mainly made up of triglycerides. So only a small portion of this molecule has those phospholipids that are surrounding the outside. An even smaller portion is cholesterol. And then the smallest proportion here is protein. So these are the least dense version of this molecule, but they are the largest because of all the space being taken up by the triglycerides. As we move down this list, these molecules are going to get smaller and more dense. So the fats that are contained within that lipoprotein structure are going to be packed together more tightly, with the most dense being our high density lipoproteins. Hence the name, high density. They are the most dense, the smallest. And so all of these are different versions of a lipoprotein, which is what we're going to use to transport these lipids to the various tissues throughout our body. So either to the muscle tissue where some of our fat is stored, or adipose tissue where a majority of our fat is stored. And then whatever is left over will be sent to the liver to either be stored or turned into some other molecule. So where or how it is used, again, is what determines the health effects. So whether we consider it a good or a bad fat. And so related to cholesterol, we're typically thinking about either low density or high density lipoproteins. You can see low density lipoproteins are 50% cholesterol. High density lipoproteins are only 17% with a very high concentration of proteins. So almost half of this molecule is made up of proteins. And so these low density lipoproteins are much more likely to essentially make a mistake during transport. And so rather than making it to their destination, they have a higher chance of sticking to the linings of our blood vessels, of our arteries, and thus depositing cholesterol there. And then that cholesterol will collect any other like cellular waste that's being removed, any of the minerals traveling through our body. And so that will start to build up a blockage that could eventually lead to a plaque, and thus increasing our blood pressure or causing a complete blockage and stopping the transport of blood in that area. High-density lipoproteins, which again are mainly composed of protein, right? 50% of the structure is protein, can actually remove any cholesterol that has been deposited on our arterial walls. Because of these proteins, they'll pick it up and help absorb it into that structure, and thus are helpful for removing any of this extra cholesterol that is within our arterial lining. And so HDLs are considered the good fats or good cholesterol, and LDLs are considered the bad or unhealthy cholesterol. And so we've made a few references to HDL to LDL ratios in this class already. And this is why that is important, or this is why that's referred to when considering the health of a food. You want a high proportion of high-density lipoproteins and a low proportion of low-density lipoproteins. So thus, the ratio of HDLs to LDLs, you want to aim for a high one. So more HDLs than LDLs. Okay. So here we see some of the examples of how these molecules are being moved. Right? So bile helps create these micelles. Lipase will then come and start digesting some of those fats, and that helps us get these long-chain fatty acids across our cellular membrane, whereas the short-chain fatty acids, medium-chain fatty acids can just be absorbed themselves. Our cells will then convert those into triglycerides and then make a lipoprotein in order to transport them out of the small intestine and start circulating through the body. But again, these fats, our lipids, skip the liver. They go into the lymphatic system, into our lymph nodes, and will skip the liver and then be deposited into the circulatory system near our heart, which will lead to them eventually making it back to the liver, but they skip that on the first turn, circulating around. That's what we see here. You see a chylomicron, a lipoprotein being transported into the lymph, into a lacteal here, where it will not be deposited into the blood until it reaches near the heart. Okay, so here's a good depiction of the size comparison between these molecules. Like I said, as we move down this list, they generally are going to decrease in size and increase in density. So we have our chylomicron, this large purple shape that you see here, right, being the largest and least dense, mainly made up of triglycerides. We then have our very low density lipoproteins here in yellow, and then our low density lipoproteins in brown, and then our high density lipoproteins in red. So again, as we move down this list, the molecules are getting smaller, or these structures are getting smaller, and the density of that molecule, of the fats that are contained in it, and the density of proteins are increasing. So with high density lipoproteins having the highest concentration of protein, and thus the greatest chance of success of reaching their desired destination, and thus being the least likely to mess up and deposit cholesterol or fats somewhere in the body where they're not supposed to be. So once these chylomicrons make it to their destination, the triglycerides are going to be disassembled by the lipase that is already contained on that lipoprotein before they actually enter the tissue. So the chylomicron travels to the destination, bonds with the tissue that is going to eventually deposit these fats into. Lipase then breaks down those fats. So again, digesting the triglycerides back into monoglycerides and fatty acids, and then deposits those monomers into the target cell. So whether that's their first stop would be to our muscles and our adipose tissue, and then whatever didn't get stored there will make it to the liver. Right? And our body uses these fats for a variety of different things. Mainly for the purposes of this class, we've been talking about their contribution to our energy. Right? They are extremely energy dense. Fats provide 9 kilocalories per gram, so the most energy dense of all of our macronutrients. And these can either be utilized immediately, and they are the preferred energy source of our cells when we are at rest, or they will be stored for later, either in those muscle cells, in our adipose tissue, or in the liver. And then all the other roles that we'll talk about in the next portion of this lecture that fats can have come from our cells turning them into other structures, so into hormones, into cholesterol, into phospholipids, which will then help us make other organelles or other cells, etc. Okay. So that is all for this portion of the chapter, and I will see you in the last video covering lipids in the next one.